Thanks very much, Daniel, for inviting me to join this panel. So these are my details. I'm going to be talking about the Seek and Blast tool, which I also talked about on the first conference day. But for those of, just as a quick recap, and for people that might not have seen this presentation, so Seek and Blast is a semi-automated tool that's designed to fact-check nucleotide sequences. So on this slide, you can see a materials and methods section from a paper, which is specifying 2 nucleotide sequences, which are characterized by just 4 letters, indicating the nucleotide bases in DNA and RNA. So we've got these strings of G, C, A, and T, and these are essentially anonymous. These are anonymous strings. Nobody, including myself, can see what they are, and so they are linked to identifiers. So Seek and Blast can extract those sequences and their associated identifiers by scanning surrounding text, and then assemble a list of facts to check. And then it checks the identities of these sequences using a tool called BLASTN, which is very widely used in the molecular biology community. It was published in 1990, and it's, you know, it's a household name. So we basically blast those sequences across a human sequence database, and BLAST provides its opinion of the status of those sequences, which genes they target, or whether they're targeting or not. And then there's a comparison between the BLASTN predicted facts and the facts that are presented in the paper. So this tool was written by Cyril Labbe, who is on the call with us. Certainly, I suppose I found these wrongly identified sequences, but if it was up to me, we would still be pulling these out manually and screening them by BLAST. It's Cyril that wrote the program and saw that we could do a lot better through a tool. So this is a link to the publication. And I'll just show you a quick example of a Seek and Blast output. So the tested file appears in this left-hand column, and there is an automated link to PubPeer. So you can post the results if you want to. We haven't used that automated link because we always check our results manually, and that's very important due to the complexity of the genetics literature. So Seek and Blast will give a nearest distance using intertextual distance analysis to a paper in a reference cohort. This paper is in the reference cohort, which is why that number is so small. It'll indicate the gene identifiers that have been identified and also the species identifiers. It will highlight any contaminated or misidentified cell lines that it identifies, and there are all the sequences listed with their various statuses indicated, and when something is not right, that's highlighted in red. So that's that incorrect non-targeting sequence that was actually highlighted in the previous slide. So Seek and Blast is freely available at this website, and we recently published a protocol which gives a very detailed standard operating procedure for using Seek and Blast. So again, just to really quickly recap some results that I showed, I can't remember which day it was, sorry. We screened over 2,500 papers, identified 700 or more with more than 1,500 incorrect sequences, and in targeted screenings, we have very high rates of papers with incorrect sequences. But in terms of just screening human publications from different journals, we found over 25% of the papers that we screened contained at least one wrongly identified sequence. And this mapped back in the case of one journal to over 10% of all original papers published in these years with an incorrect sequence. So I also talked previously about this computational research integrity kind of ecosystem. And so I'm just going to run through, I guess, challenges and opportunities for some of these different groups. So I'm going to start with the target features. These are the things that tools are developed around, and the tool developers. And I've always wanted to talk about this in a talk, so here I'm doing that. Donald Rumsfeld's view on knowledge around known or unknown unknowns, and I guess it's the kind of the known unknowns, the recognized gaps that represent opportunities for tool development. But the most exciting, of course, are the things that we don't know about. And I spend a lot of time thinking about this. It's obviously hard to work out what you don't know. That's probably where the really big opportunities are. So in terms of the people that I'll refer to as the target spotters, I guess the opportunities are, you know, the capability of matching people that might have a new target with someone who can develop a tool. I guess we could do that in different ways, you know, competitions, idea pitching, exercises. Essentially, it's about matching people with common interests, but with very different expertise. And I think that's essentially what led to the development of Seek and Blast. And these people who are target spotters can go on to become research users of tools as well. In terms of challenges for tool makers, look, I'm not really the best place to make a comment here, but I sometimes wonder whether we try too hard to aim for perfection. I recognise that humans use a lot of imperfect tools all the time. I was thinking about a good example. I came up with a knife. 2 days ago in the kitchen, I used a knife to cut an Easter bun and I cut into my finger. So knives are problematic tools, but I still use them. I think there's an opportunity to learn, what's good enough for different applications, recognising that that's not a one-size-fits-all proposition. We need to consider risk-benefit estimates, particularly around the value of false versus true positives, and those values will differ according to application. So now I'm going to talk about the research users of tools. Here, I think the challenges are essentially lack of awareness, time, and a degree of I don't want to know, which of course exists in some quarters of the research community. We need to teach, can't actually see this slide of mine, but I think if we teach literature appraisal tool techniques, that can lead to tool use, and we can also teach tool use. I'm about to do that this morning at my university to undergraduate students. There's also the perception that journals and publishers won't respond. So kind of what's the point of using tools? I think that's, obviously not always a correct perception, but unfortunately at times a correct perception. So we've proposed simpler ways of reporting issues to journals and also faster and more neutral post-publication responses in the literature. So finally, I'll talk about journals and publishers. A couple of things that I've heard during CRI-CONF has been challenges such as tools really need to be easy to use for journals and they have to integrate with their existing workflows. So that's clearly very important. And also this, we've heard a couple of times, I think, the more we screen, the more problems we find. So some of the opportunities I think are obviously partnerships with, companies, researchers, particularly around tools that need human support, and recognising that journals don't have to do everything on their own. And there's opportunities for prioritising in terms of how tools are applied. They can be applied in an easy way to identify the low-hanging fruit. And they can also be prioritised in terms of the urgency or the impact or the risk of the problems. And we're starting to do that in terms of our citation analysis, prioritising papers that seem to be closer to the clinic. I'm going to finish just with a couple of other slides. So on my first talk, I talked about cancer. I'm going to just briefly talk about climate change. Essentially, the approaches are mitigation and adaptation, and those things seem to be relatively separate. Mitigation is largely about structural and systemic change, and I think we can argue that changes to the literature also require changes of this magnitude. But I think in the meantime, either while people are waiting for this to happen or in case it doesn't happen, people that use the literature really need to adapt to the new reality, and that is that many parts of the literature are not reliable at all. And again, I will be teaching that to my students this morning. A final word, I think I talked about risks in my previous talk, but perhaps the biggest risk really is that people eventually might stop listening to us. Researchers, journals and publishers, we all occupy very privileged positions in society. We ask governments and institutions to commit to solving difficult problems. I've given examples. Of course, climate change is probably the most important. And I think we need to tackle and importantly be seen to tackle our own difficult problems. So I'll stop there. Thank you.
Oh, thank you very much, Jennifer. So what about you, James? You go next.
What about me? Good looking. Let me do exactly the same screen share business here. Okay. Are we seeing a rather boring slide that looks a lot like a presentation opening? We got it? Okay, let's rip through it. I love Q&A. I love conference sessions where people get to ask questions and throw rocks at me. So I want to get to that as soon as possible. I'll introduce you to everything that we're about here. And we'll get to Amit. Side note, great pleasure for me to be here with two people whose work I've actually read closely. Doesn't often happen at conferences. It's usually people I've no familiarity with whatsoever, and then I have to catch up on the fly. So good job, Daniel, and everyone at the conference for putting together a thematic session that actually makes sense in context. Okay, error detection tests, the 5-minute version. This should be fun. That's my job. This on the right-hand side is an engineer being, well, sort of mildly tortured. The thing he's wearing on his leg is what we build. It's a cloth-mounted wearable monitor that is designed for measuring human movement and biometric stuff. It has a lot of, it's primarily a healthcare device. It's a medical device technically here. It has athletic training uses, general uses. It has industrial applications, military applications, etc. I'm the chief scientific officer of a company that builds that. My other job, this is my other place of work. That's a 3-seater couch. That is where I do this job and where I've always done this job, except for the brief interludes where people have paid me to do it. Like most people who got interested in this, it became a passion project very quickly. What is that job is reasonably straightforward. This is from a paper I'd finish if I didn't help run a startup. I'm the chief scientific officer. Other people shake hands with investors. I make sure our stuff works. What is an error? Strong inconsistency or impossibility within the contents of a scientific paper. Beyond that, it's incredibly variable, incredibly. But the ones that I primarily deal with are computational. So what you're about to see is not maths that will shake you to your core due to its complexity. It's the exact opposite. All this work and all the time that I've spent on this work has been trying to couch within the boundaries of academic reality extremely simple computational observations. It takes a lot longer than you think it might to formalize and observe something like this and then turn it into a test. That being said, when you actually get to the point where you've built a computational test and you can look at a scientific paper and say that result presented there is either vanishingly unlikely or completely impossible, you get this stuff. I'm going to talk about 4 because these are finished. You can read about them. All of these are with the erstwhile Dr. Nick Brown. We started when I was just a doctor and he wasn't, back in 2015. And we're still chewing through things gradually to this day. But he's retired. And as I said, I work for a company that means I don't get a lot of sleep. So let's tear through them real fast. Grammar is extremely straightforward. Any arithmetic mean is when you collect data in whole units, an integer over an integer. When you have a low n, a low cell size or a low sample size, it means that many of the decimal components of that mean, x.xx, the last 2 x's only have a series of restrictions on what they can be. So here's a nice example. Let's say we're measuring integers in a sample of 17. We add up all the scores. Obviously, a lot of these will be 1s. We add up all the scores. We get 20 for our numerator. We divide that by 17. The mean is 1.18. If we get 21, one of the scores increases. We get 21 over 17. The answer to that is 1.24. So if in this particular environment, someone reports a mean and it's from 1.19 to 1.23, it is impossible. None of the values in that range can be built out of 17th units. This works obviously very well at the low end because there's lots of gaps between the individual decimals. And then as the sample size starts to get larger, there are some interesting interrelationships between that. Let's not do the maths, let's keep going. So I told you it was going to be simple, that's simple. Okay, he said immediately moving to a slightly more complicated one. Sprite is sample parameter reconstruction via iterative techniques. Techniques plural because there's more than one way to do it. Basically, if you have a description in descriptive statistics of a distribution, hopefully the range, the minimum, the maximum, mean, SD or SE, and N. You can take the values that you assume go into something like that, give them an appropriate decimal, and you can shuffle them, basically. You can shuffle in how the variance is distributed. a number of algorithms that you can use to do that, or you can use a simple decision rule. You can use one up, one down, which is what we originally did with Sprite. What it means, though, is that you can generate a somewhat plausible distribution depending on how you, well, another box we won't open. What it means is a lot of the time that the distribution that hides behind descriptive statistics can be immediately apparent. So you can't see the numbers on the right-hand side, but that is a simple, maybe a Likert scale kind of sample. An N of 45, integers between 1 and 7 for our answers, mean of 3.53. And all I've done from those two, from a Sprite tool, this is Nick's one, is change the standard deviation from 1.56 to 2.56. Now, the 1.561, there's very, very many distributions that could potentially be. So we can't really tell anything about it. But you make the SD particularly large or particularly small. Particularly small, it ends up being incredibly narrow. Particularly large, it ends up being bimodal, obviously, with the modes being at the extremes. We found a whole bunch of papers with standard deviations that are too large to be realistic. I'm never quite sure why they're there in the first place, but you get to a certain point reconstructing these distributions where you can say that data looking like that is unlikely. Third tool, this one's really straightforward. Anyone who's familiar with a big table of values that goes into a regression analysis will know that a lot of the time we code binary human variables. You see these in regression tables a lot, male, female, high income, low income, rural, urban, anything like that that's coded as 0 and 1. Anything that we have a mean and a standard deviation of 0 and 1, the mean and the standard deviation are mathematically determined. If you have a mean of A, you always end up with a corresponding standard deviation of B. It's really, really simple. What it means is that if you have binary data, an N of 50 and a mean of 0.34, the standard deviation is always 0.479, full stop, end of story. So if you see a table of regression values, and that is not the case, the data isn't right. Full stop, end of story. Last one, just for fun, rivets. This one is a little bit strange, but it's actually designed to detect not so much an error as the presence of hand calculation. Now, this is really interesting. This is the most recent one, and we found some of the most interesting results in the broader literature with it. So when you see something reported to a certain number of decimal places, generally you don't know, for instance, let's take this first sample that I've invented here, 4.53. That 4.53 is technically somewhere between 4.525 and 4.5349 repeater. Now, if you take that condition and you put that over any of the means and any of the SDs, the eventual statistic that you get actually has a fairly substantial range. And in this case, it's between 4.09 and 4.23. Now, bang in the middle of that, what you'd expect if these decimal tails were truncated is a particular t-value. And this holds for any other statistical test that is a basic NHST framework, anything. ANOVA, regression, z-test, doesn't matter. So if you get 4. That is the number that you would get if you had truncated all the decimals. Now, if I go through your paper and you say, I have calculated this from statistical software, and every single one of your values is truncated, then it's possible to say that it's incredibly unlikely you use that statistical software, maintaining a decimal tail, usually to about 16 digits, to be able to do your calculation, or there wouldn't be that pattern in the actual test statistics. That one's a little curly, but it's really good fun. There's a whole bunch of other stuff I'm not going to tell you about. There's associated work by some related researchers. Some of it's really interesting. We have a consolidated website for all of these tests in the same place it's being built. There is some work going into making some of these tests into browser tools. And I have a whole bunch of unpublished results that I really don't have the time to tell people about responsibly. One is a Sprite variant that works by setting a distribution rather than just shuffling numbers around. And we have an unpublished paper on the parameters of what we call selective redaction, which is basically saying if you have two samples, how much data do you have to throw away? We either treat as an outlier or treat as an outlier to be able to engineer. So that's a 9-minute introduction to error detection, and I hope that gives us some context for what's about to come.
Excellent. I love it. Very, very, very cool. All right. So next is Amit.
Okay. Hi. Good afternoon, everyone. And thank you, Daniel, for organizing it and for inviting me for this. Let me share my screen. Yeah, so it's a very interesting meeting that you have. From a computer science point of view, this is a topic that that we started working on when we were part of the media forensics program from DARPA. That was not focused on scientific publications. It was mostly on images and identifying whether images have been manipulated. And from there, there was this separate program that the Department of Health and Human Services had, and I submitted a proposal to that on whether we could use it for identifying image manipulations in scientific publications. And that's how this started. The project is almost over. This is the last quarter in terms of funds. We are almost over from in the project. But it was a struggle initially, and I will get into why it was a struggle, especially working in an area where, which is kind of new, and you are trying to build up the area. It's never easy. So I will get into some of the results that we have obtained, and then I will discuss a little bit about the challenges. My student, Ghazal, she presented a more detailed version of what I will talk about on Monday in a paper. I'm Amit Roy-Chowdhury. I'm a professor in electrical and computer engineering at the University of California in Riverside. I also hold a cooperating appointment in computer science, and I work in computer vision and more broadly in machine learning problems. The particular project that I'll be talking about is on identifying duplications in images in scientific publications. And as I'll mention later on, one of the biggest challenges in this domain is how do you get data sets to work on? So we collaborated with Elizabeth Bick. Many of you might know her from the data set she created. And so she has gone through a large number of papers, 20,621, where from there identified a number of images that had at least a figure containing inappropriate duplications. These were done manually, and they provide a ground truth for us in computer science to identify or to evaluate how well our methods are working. And that is essentially what we focused on. So given this data set, can we develop a computational method that would be able to identify these image duplications? And if we are able to, how well does our method work? Right? So that is essentially the main goal. So given the data set containing this, we want to learn a model that would be able to classify a pair of test images to be either copied or not. And our specific contribution here was that we came up with this approach that relies on image extraction, a region of interest generation, and then detecting whether there is a copy-paste duplication. And we came up with a particular machine learning method to do this, and it led to significant improvement in accuracy over existing methods, about 13% improvement. So the framework that we had has the following steps. First, we need to extract the figures out of the papers. So I'll go over the steps of our method, and then I will talk about what is really difficult over here and what is hampering the development of work in this area. And it would kind of blend into the first question that Daniel had in the email he sent yesterday, in terms of the two strawman examples that he set up. So extracting the figures out of the papers, that is the first step. So if you are given the papers, just getting the figure regions itself. And remember, it's not just often that it's just the figure. There is often figure and there is text embedded into the figure. There is a table embedded into the figure. And you need, and if you're trying to identify manipulations in the actual image portion of it, you need to separate that out. And that by itself is a non-trivial task sometimes, depending on how the figure is set up. And that leads to the region of interest generation. So you separate out the text, the graphs, the charts, and you focus on, this is the part of the image, or this is the part of the figure that I really want to focus on. And then you get into the duplication detection method. So the particular approach that we proposed for this was using a Siamese network. I'm not going to get into the details of that. So Ghazal presented this on Monday, and that is more for a more detailed machine learning kind of discussion as to why a Siamese network versus anything else. But let's just abstract it out that we come up with a machine learning method to do this. So the way this happens is, if you look at the figure below, so you have the input from where you get the raw figure, and you can see from here that actually, this way. From the raw figure, you can see that there are these tables, charts, and there's the images. And the interest, the part of interest, the part that is of interest to us is this portion over here. I'm assuming you can see my mouse. So is the portion over here. Once you get this, you need to analyze this. And now you get into the typical computer vision image processing problem. So the first is segmentation, identifying these regions. So how you get rid of the black regions and you just focus on what is the informative part, right? So that is the basic segmentation problem. And then you get into the machine learning problem. And this machine learning problem, what it does is, given a set of training images, so you take the entire data set, you separate out into a training set and a validation set. On the training set, you take the examples, you take the ground truth that these are duplicated, or these are genuine and not duplicated, and you train this machine learning system to classify whether a pair of images is duplicated or not, right, based on the ground truth. So basically, you've told the machine learning system, here are two images, these are pristine, benign images, there's no duplication. Here are two images where there is a duplication. And you do this for a number of images, and then the system learns something about what duplication is. And this particular approach that we had, the particular learning system, was a Siamese network. And the reason we did this was twofold. One is, you are training these pairs of images. And the Siamese network is ideally suited in this kind of a scenario. You give two pairs, they're either a duplicate of each other, or there is some duplication in them, or they're not. And the Siamese network is well suited for that. The other thing is, the Siamese network, as compared to many other deep learning networks, require less amount of data to train them. And data, as I mentioned in a minute or so, is one of the big constraints in this kind of work that we have. Okay, so so our task was to go through all of these. So figure extraction, the ROI generation, segmentation, the labeling, as I mentioned, and then number of the pre-processing steps. And then we evaluated this method versus a number of other methods that have been used for duplication for duplication detection. And we get a pretty significant bump in performance, depending upon how we do the pre-processing. So compared to, for example, the closest alternative, which was a 13% improvement in accuracy on that data set that we received from Dr. Beek. So this was a summary of work that we have been doing mostly over the last 1.5 years. And before that, it was largely to get all the pre-processing and data in place. And that leads me to the last slide I have. And I want to focus on this to some extent. So I identified 3 big challenges in working in this area. Like if my student had to continue working on this, or if I had to have another student do her PhD on this topic, what are the big challenges? One of them is the availability of data. So in order to publish high quality papers in computer science venues, especially related to machine learning, they're very, very data dependent. Obtaining large volumes of data over here is a challenge. And I know, Daniel, you didn't want to discuss about the points that you had, but I will take the liberty of saying something about the first point you had in their email yesterday, if that's okay. So you had 2 strawman examples. One was that computer scientists might think that this is a very easy problem and what's the big deal in it? And on the other hand, biologists who are creating the data might be wondering as to whether we can have automated methods to do this. I think a large part of why this problem has not, or computer scientists may not be aware of it, is because we do not have good and large-scale data sets on which this problem can be well defined. And that is a major issue. So Dr. Bick has done a great job, and she has helped with creating this data set. But if you think of the amounts of data that machine learning problems work on, this is as a percentage, it's still very small. And we do not really do not have the kinds of data sets where we can enunciate the challenges very clearly. I think it's a very hard problem. Having worked on it for 3 years, I think it's a tremendously hard problem to do this in an automated manner. And we have to be able to represent the problem properly. And that leads to my second point, which is the challenge of pre-processing. It is probably possible for those of you who specialize in identifying these kinds of manipulations in publications to say, here is an image in a paper that we suspect is problematic. That is a good input for a computer scientist, but there's still a lot of work in terms of pre-processing that input and bringing it to a level where we can analyze it. First of all, you have to get rid of all the text. You have to get rid of all the tables and charts, and you have to be able to focus on the right problem. I mean, each of these in computer vision or image processing are difficult problems in their own right. In fact, this is the whole problem of region of interest generation or saliency detection in an image. What is salient? Like if you look at any of the images that are showing up on the Zoom gallery screen, if we were asked what is the salient region, we'd probably say it is the person, like each of our faces. That is a salient region. So there is something that we know fundamentally, that in this video conferencing setup, the face is the salient region. Given that image that you have in a publication, how do you identify what is the most important region to focus on even, right? And if all of this was identified manually, and then that is a hard task by itself. Who does all that work? So that is where this problem becomes so difficult. And I think so identifying well-defined computing problems has been a challenge. And I think the reason why some people might think this is an easy problem is because we've probably failed to define precisely where the challenges are and what the difficult problems are here. So with that, I will stop and we can get into the panel discussion.
Thank you, Amit. You can swear that I didn't invite you after my competition, but I had a competition presentation, but I was talking about kind of similar problems and trying to propose a way of trying to solve that problem through a competition where we'll create a data set. So if I might allow like indulgence, because I'm, I guess, a chair of this panel, I'd like to talk a little bit about the stuff that I've been doing with the tools. I hope that's okay. So I'll share my desktop. Right. All right. So I developed a tool for detecting reuses in my view, my honest view, is far simpler than the tools that that Amit is talking about, because he's talking about learning what makes something similar, and what Ed talked about yesterday. What we were aiming for with this tool was for scalability. So basically uploading a PDF and comparing it across hundreds of or millions of PDFs. So in that sense, the false negative rates must be massive. So, but what we did was we recover cases, and then we ask a panel of people to rate them, whether they saw all the range of data that each paper had, the caption and everything, the full paper, so they were able to see the information and label them. And we found that it seems that across papers there are more issues than within papers. So that's kind of it's a motivation for us to continue with that approach. So now we're trying to scale this even more because we have this preprint, but it's not really that scalable. The scales within the same first author and last author, but now we're moving into basically one PDF match against everything. Now, what we've developed is it works regardless of many transformations, and we use a standard technique from computer vision called key points. So it's able to capture uses, even though there could be modifications. There could be blurring, contrast changes, rotations, and things like that. Now the tool is available, and I hope to discuss this, and maybe you have your own experiences how to work with universities, and hopefully they won't watch this, the lawyers, especially from Syracuse, but it's been the biggest pain ever, and just giving access to this tool to editors and companies especially, it's just been very, very slow. But we have a tool, it's available here. It's called Dr. Figures. I hope you get the joke. But, and this, the funding for scaling this and making it available was also part of a grant from ORI from 2018, our first attempt at ORI. But that award ended. And this tool is web-based, so just uploads, now it allows us to 50 PDFs, but there is no really limit, but you can upload it and it detects reuses. Now it's not smart enough to, well, it detects some reuses, and this is a paper that was retracted, so it's okay to be to be filming this and recording it for later. But we realize that low contrast areas is not that great for detecting Western blot, for example, and sometimes some areas that have text and graphical components, it detects them as reuses, but they're not really reuses. These are okay reuses in science, so there is no problem with this. We also added some feedback that's to the for research integrity officers, that they usually apply some kinds of filters in Photoshop. So we try to reproduce those filters. So if you click each of the reuses, it will show this preview of equalized histograms, false colors, gradient maps, and things like that. Now, one of the complaints that we heard a lot from research integrity officers was the idea that of how can they make any decision with this? Yeah, there is a reuse, but sometimes there is like uncertainty about that reuse. Like, is there a way of giving some numerical representation of how good or bad that reuse is? And what we propose with a student who left, and he's actually now at Purdue working with Ed Alan, was to try to compute some sort of p-value. So basically, what are the chances of a reuse happening at random? And here, as an example, I'm showing you 3 kinds of reuses. So you can have text reuses, you can have Western blot reuses, and you can have something like a tissue reuse. Now, it should be relatively obvious that text reuses are very common because you use the same font, the same color. So basically, the probability of that being reused should be higher than something like a picture of a tissue. So we develop a method to compute a log probability. So basically, you will see these numbers, and we didn't choose any thresholding, but this is a feedback, a number that we could give to research integrity officer, and the person will be able to say, yeah, that seems to be below a certain threshold, and maybe that's constitutes some evidence that they can use in their investigations. Like it's like seems to be very, very unlikely that this happened at random. So here we plot a histogram of things that we discover, the analysis that we did for different kinds of images, and you can see that text images are more likely to happen at random, reuses are random, blots are less likely, and then even less likely than that are microscopy images. And this is also was part of another grant from ORI, where we try to develop techniques for giving feedback to research integrity officers and thinking about the human in the loop of these investigations. And the last project is about detecting manipulations. And this is an ongoing project. So this is part of the same grant. And basically we're looking at trying to learn from a vast set of data sets, try to understand how the noises usually work on those data sets. So if there are manipulations, generally the noise structure, the statistical structure changes. So we're trying to detect that through computational methods, and we try to make it fast too. So again, I'm trying to focus on scalability here. These techniques, in all honesty, they are not as advanced as things that people use these days, like deep neural networks, but because they're intended to be really, really fast to use. And we have some decent performance when compared to other standard techniques that are applied to general images. And with that, I'll finish my part of the presentation and go back to the panel view. Okay, great. So I had questions prepared for all of you, but I couldn't help but think about that each of you individually, their main career is really in a different area. Well, at least for Jennifer, I think today you revealed that you kind of are left, well, yesterday you revealed that you left some of the more standard research that you used to do. But in a sense, I would say that that you were that each of us is doing this on the side, or used to do it on the side. So there is no like a lot of incentives to do it as a main, or there is not a lot of reward for doing it as the main part of your research. So can you say something about that? Like, why do you think that is? I mean, there could be things related to funding, could be things related to no venue. I mean, I remember Amit telling me, you know, it's kind of hard to find venues for publishing this kind of work. And yeah. James, if you two, you kind of, you mentioned that you work in a company, but maybe you were doing this before more full-time. So please, let's expand on that point. Let's start with, I guess I'm going to go in reverse. Let's start with Amit.
So in terms, so Daniel, can you repeat what is the question you want me to?
So the question is, it seems that each of us is doing this research on the side. Like it seems that your main research area is something else.
Yeah.
Why is that?
Why is that is largely a question of history. This is a relatively new problem to be able to identify image manipulations, and my main research has been more on video analysis and now more on machine learning. This I look upon as an application. But if you are asking why is it I haven't put in more resources into this problem, I think the reason is the challenges I mentioned, that where is the data and what are the precise problems? I have been struggling to define these two, and I do not want to put a student in it who has a hard time publishing in top tier computer science venues for his or her PhD. That has what has been the hesitation.
What about you, James?
I would say two things. One, I agree with Amit completely in the fact that people have approached me many times about how exactly are we going to turn these rather manual tools, James, into something that is scalable. As someone who works with a company now with half a dozen people spread across different aspects of front and back end UI software, firmware, I can appreciate that if you could get the data out properly, just how easy it would be, especially with our research, which is reasonably straightforward. And the center of the problem is the thing, if you gave me a magic wand, I'd kill the PDF file. I would make a paper, basically a big bag of XML data where images were capable of being retrieved by themselves, where nothing strange came into text, and where optical character recognition behaved itself properly, and everything that is in our scientific archives and the sort of sum total knowledge of empirical history was put into exactly the same boat. But there is no real genesis. I mean, this becomes a standards and practices issue. I'm trying to get people excited about that. I mean, it's like a wet afternoon in Surrey. And would you rather be on the beach in Spain finding excellent new findings and being novel and having flair, or would you prefer to talk about standards? The answer to that is inevitable. So I'd love that, and I'm sure it sounds like something Amit would welcome. It just also, I mean, it's never going to be a priority. With regards to the issue of the sort of spare timeism, well, let me put it this way. There's no journal that is with a specific interest in scientific error or error detection or anomaly detection or anything like that. There are no major funding mechanisms that pursue this, especially on a program grant level, which as far as I'm aware, there's zero. I've never heard of anyone who does work in this area getting a job that was at the sort of postdoctoral or early career level that was directly related to doing this work. And you can't even, when it comes to, does it actually have a scientific sort of front-facing credibility when you do it? No, I've spent 4 or 5 years chiseling away at individual cases, and eventually something gets retracted 4 or 5 years later. It can take that long. Other people have worse stories than me, I can tell you. And when that happens, no one will say your tool was directly responsible for finding its result that never should have existed for whatever reason. They will write a retraction notice that does not mention you or the tool. As in, this literature was corrected because reasons. So the only way out of that particular conundrum is a lot of time, and someone who works at a policy level somewhere who takes a particular interest in this and making as much noise as possible. But fortunately, that happens to be my specialty. I'd like to hear what Jennifer thinks.
Actually, I can see someone's got their hand up.
Yeah, actually, Jennifer had a paper on this issue of PDFs that someone was mentioned in the beginning.
Yeah, that's right. That's actually some work that Cyril and Guillaume Cabernac did that I was associated with, that was published in Scientometrics last year. Look, I'll just quickly sort of, I guess, address the question. I guess, look, I'm like everybody, you know, you do research in combination with other things. So I've got a day job and I do research on biobanking and its support of medical research. But, you know, we've had sort of more success recently. We've been funded by ORI. We've also got, I guess, the equivalent of a NIH grant in Australia. And I think we were able to get that because we could make the case that errors in the literature affect research that impacts human health. So it's good to see a funding organisation listen to that. I guess I certainly agree with James. I think there's an under recognition of the importance of errors in the literature, but the absence of a dedicated journal in a way can be a good thing because it gives you the opportunity to publish in lots of different journals. And I think that can allow you to reach lots of different audiences, and that's important. You know, the only issue with specialised journals is you can end up being a little bit, you know, preaching to the choir. So certainly the people that like errors, great, yep, we'd all read those papers, but we need a lot of other people to read that stuff.
Yeah, I think I'll just start taking questions from the audience. I don't know how you feel about it, panelists. Yeah?
Well, that was why we were here.
Yeah, okay, good. Now there were Ed is with his hand up. So let's see, Ed, what you have, and then I'll go back to read the chat.
Sure. I guess I disagree with some of the things that were said. I mean, number one, as far as anybody funding research in this area, certainly DARPA is continuing to fund research in this area. The new DARPA Semaphore program, which we're part of, is continuing specifically to look at documents. We are continuing looking at this. I presented yesterday our end-to-end system, which actually has a very good PDF parser. It's a very good PDF parser. It's probably one of the better ones out there that will take a PDF apart and split up the images and the text and binds everything together. As far as people getting hired, one of my PhD students has been hired by Apple, their new machine learning group. They stood up in Seattle, and she's specifically going to be working on documents with Apple because Apple's very interested in this. So I think we're going to see more and more interest in looking at this whole idea of documents, document forensics. And I think there is a problem with data. You know, we're certainly collecting more and more data and trying to expand it out. But, you know, we are continuing to work on this problem, both with DARPA and ORI. And I see there's more and more people working in this area. But I think that there are people looking at this problem now, and this problem is part of a larger problem, just looking at documents in general and how documents can be falsified, not only looking at the text, but also looking at you know, the images and other information that might be embedded in the document. So I think there's a lot of people sort of working in this area right now.
So I think I should clarify something. So I did not, when I said that about the problems, I did not mean document analysis in general. I meant just in the scientific publications, detecting error or detecting manipulations in scientific publications, which was a specific focus of one of the projects we had. Just in that one, we had a hard time collecting or getting the right kind of data.
Yeah, I agree with you, Amit. However, I think, again, looking at particularly at scientific publications, again, you know, we are continuing to look at this with our funding from DARPA. And I think we are collecting and creating a data set that, you know, we hope to be able to release soon. It's not anywhere near the size we'd like it to be. But I think there is an interest in this, in many of the manipulations that you see in scientific documents. We are beginning to see in other types of publications, other scientific documents, not just related to the biomedical area, but we're also seeing these in publications in chemistry and in other branches of science. And, you know, we're interested in all of those, and we're going to be looking at those types of things. So I think you're going to see more people working in this area in the future.
Okay. Yeah.
Anything from the panels? So let me just say that several people are asking about this awesome PDF parser. One thing. The other thing is that people are interested in your paper, Jennifer, that I just mentioned about how PDF is bad. I also had problems parsing PDF, so I'm also interested in this.
So
this tool.
we're writing a paper
also
right now.
Yeah.
And when we get it written and submit it, we'll put it in archive, and I'll send you the link, Daniel. You can send everybody to attend at the meeting. And we hope to make parts of our code be available, including the PDF parser. So
cool. Also, the DARPA is very specific. It's like a very brutal process. I feel like it's just like, yeah, I was thinking more like just basic funding for the tools, for example. Like, why can Jennifer get a grant, or maybe she has a grant, but I will say, even though they're being very generous with me, they have like a certain way of doing things that is kind of short term. So they're trying to improve that. So it's not like NSF also doesn't fund tools that often. You have to kind of justify it from the scientific point of view. So that's kind of what I was referring to. So one question that I wanted to ask you is, and Jennifer, I'll start with you. Have you seen people adapting to your tool? Have you seen like I feel like we discuss guns and we discuss these very scary things happening in the world, but have you seen anything where fraudsters are kind of saying, oh, this is how I need to modify my genetic sequence so that it doesn't get caught by your tool?
Daniel, I'm very glad you asked me, because I have actually prepared an answer to that. Look, I think obviously adaptation is possible, and we're actually looking for evidence for that. That's probably the best way to do it. We're looking at whether the types of nucleotide sequence errors are changing over time, particularly in the context of journal screening. I guess as a peer reviewer, I see papers as well, and I have seen papers where, for example, sequences are just omitted. That's the easiest way, isn't it? You just don't put the sequence in. You just say, oh, we got this sequence from a company, and that's kind of none of your business. So I think there's a role for journals in terms of enforcing requirements to specify reagents. That's good for everybody. That's just good science. Look, I worry that inevitably, if we're talking about paper mills, that paper mills will learn to get sequences right all the time. They get them right a lot of the time. They just don't get them right all the time. And so that window of opportunity, may close. There will still be errors fixed in the literature, but in terms of going forward, I think that is a concern.
So James, I know that Michelle, who unfortunately couldn't attend this meeting, developed also things related to statistics, and she's releasing her tools out there. So do you think people are adapting to the analysis, the kinds of analysis that you run?
No, definitely not. I'll tell you why. There's a thin end and a thick end of this wedge. The thin end is a lot of people who can't, to coin a phrase, write a scientific paper and chew gum at the same time. We start off with the thin end of error detection is things that you don't need techniques to be able to spot. I mean, we found a paper once where the same sample was described with 3 separate ends on 3 separate pages of the paper. And it's because they just kept presumably redacting the data until something actually worked. And then they actually didn't manage to go back and do their own editing. There are plenty of bad scientists. And if people are too lazy to add up, they're almost certainly too lazy to go through and predict what the outcome of a distribution reanalysis would be, or to so there's a huge undifferentiated, and so look, there's plenty of bad scientists hidden in what are allegedly good places. That will never really change to any great degree. There's a certain amount of people who don't think anyone's looking, so they're never going to change anything they do, which is just a sort of fundamental tenet of human nature. At the other end, there's if you ask more or less anyone who is either on the panel here or in the audience here, make data look real for something. Now, you could probably find some irregularity eventually, but I guarantee you that the vast majority of alleged questions answered by the vast majority of alleged data would be, if not completely undetectable, then within a kind of a margin of error where it would be inappropriate to ask questions about it if you saw it in the wild. So in general, like basic error detection catches people who are very bad at at not making errors. Look, that's the reason why when you aggregate the figures, that it's scary. These are the things that really shouldn't exist. When we found the first result in GRIM in 2015, it was so trivial. I think the first spreadsheet was literally called primarymaths.xls because we were just fiddling around in Excel. And then we realized that there was a relationship here, and we formalized it and did a bunch of, frankly, undergraduate mathematics. And then said, someone's published this. This exists somewhere. It's too trivial. It's too straightforward. And it took me 2.5 years to find a reference to it anywhere in the margin of a paper from 2006 that was about training peer reviewers to accurately measure medical journals that had presupposed this technique that no one paid any attention to for the subsequent 11 and a half years.
What about you, Amit? Have you seen any adaptation? Do you hear me? Okay.
Yeah. So can you define a little more, Daniel, what do you mean by adaptation here?
Well, there were, like, let's say, a lot of very scary examples of, GANs being used to produce images, for example, Western blots. And so the same you can say, people, let's say if we release your tool, if the people will start modifying things and to make it less detectable. Have you seen something like that, or what do you think about that?
So not in this particular application domain, I have not. Not in the domain of scientific publications. But in general, with GANs, I have, yes. In fact, we did some small work, but that was mostly on images in general, not in this kind of application domain.
I see. So I'll ask another question related to what I send you. Do you think, and this is something that I discussed with some publishers, do you think there is a case to be made so that a paper gets automatically flagged and perhaps rejected if it doesn't pass one of your tests? What about you, Jennifer? Is there any instance in which we can have like an automated reviewer that reject a paper because it's just too?
Yeah, sorry, did you want me to go, or?
Yeah, you can go, Jennifer.
Oh, thank you. Look, I think in the question that you sent, you gave us a figure of 99% accuracy. So if that were the accuracy, I would say absolutely yes. The reasons are that rejecting manuscripts happens all the time. When people submit a manuscript to a journal, you're looking at a probability of being rejected that ranges from maybe 30 to 90%. So rejection is something that, authors accept. It's not a big shock. It's not welcome. I think that if automated tools are used to reject manuscripts, there should be the opportunity of appealing for that 1% that's incorrect. But beyond that, look, I think, such a tool would be probably far more accurate than a human. I'm sure editors-in-chief, and I am one, reject papers, we're probably, at best maybe 50% reliable. So 99% is pretty good.
What about you, James?
You have to be amazingly careful. Again, I'll tell you a story because this is fun. One of the papers that we found with the original Grimm test, we could not make head nor tail of a table 3 in a particular paper that we read. And we found it, we turned it inside out, and we said, this is obviously not consistent. We wrote to the author, who very promptly wrote back to say, no, I'm absolutely certain that it's fine. I said, oh, it can't possibly be. And I said, are you absolutely sure? Yes, they rechecked. I think it was a German gentleman from memory who was fantastic. If every author was like him, we'd live in a very different world. Turns out we hadn't noticed the fact that not only was the N split somewhere in the text into subgroups that were described, so it didn't flag as a number, but there was a sub-analysis within that was in a footnote. And you put all those pieces together and you understood how it actually happened. Turns out that he was perfectly correct and we were wrong. Now, something like that happens even when you put human interest into how to put together the context of how things are reported, because method sections, as we all know, are always perfectly easy to interrogate. And in general, people describe everything in beautiful, clear, straightforward academic language. I mean, that's our hit rate, about 80%. So I find it very hard that outside of certain contexts, unless you have a standardized table, for instance, that's laying out the context and a particular column of a particular output or a particular test statistic, then it's possible. In the general sense of, can we pass this? Can we process it? And then can we turn it into something that will affect a publication outcome? It's too it's too stochastic for people to accept. I think there's a practical barrier as well as a computational barrier there. So I don't think it's going to happen. I'm also, look, there's a degree to which I don't care a great deal. There's most papers are read many times before someone notices an error. We found an error in an old Brian Wansink paper in table 1, where there were two submeans A and B that added up to a collective submean of A plus B. And none of the values were right in table 1. Now, we found this on ResearchGate, and it had been downloaded 1,600 times. Apparently, no one had ever pointed out that table 1, which only had 9 numbers in it, we don't know whether it was 6 or 3 of the numbers that were wrong, but they were wrong. And they were wrong in a way that you learn when you're 7. And I'm more concerned about 1,600 people reading it and not either having the ability or the access to be able to do anything about it than I am about automating everything that I've tried to build.
What about you, Amit? What if you had 99% accuracy in detecting? Would you just reject without the editor doing anything?
If I had a 99% accuracy detector, yes. Do we have a 99% accuracy detector? That I have my doubts on.
Okay. All right. So that's a perfect end